Okay, thank you, Katia, for inviting me to give this presentation. So today my presentation is entitled Contribution of Optical Trait-Based Remote Sensing to Ecology and Agriculture. So it will be relatively broad, and I will basically start with a few information about physical modeling, and then I will try to give a view of more applied results we had in our lab. So just to start with a bit of context, so the erosion of diversity, biodiversity, and ecological disturbances are increasing, and we need operational methods to monitor ecosystem dynamics through space and time. So here are a few examples of some pressures occurring on vegetation. So it's mainly on natural ecosystems, but also on managed systems like forest plantations, and also on agricultural lands. And so basically here, remote sensing can provide information contributing to monitor complex systems at various spatial scale, from local to regional to global scale. It allows also to link Earth observation with ecological knowledge and climate data, to feed regional, national, international statistics on diversity and ecosystem degradation, and also to fuel higher level models and analysis, integrating ecology and socio-economical perspectives. So how can we use remote sensing to actually meet the current environmental challenges? So today there's an increasing availability and accessibility and maturity of methods and sensing technologies. So we actually have a lot of new original inputs for applications in both ecology and agriculture. So we have optical sensors covering spatial, temporal, spectral dimensions. We have a bunch of new methods filling the gap between remote sensing data and thematic information. And we also have models describing complex systems with increasing accuracy. So in my presentation title, I was mentioning optical traits. So here, I will try to give you a definition of these optical traits, and I will borrow it from a paper from Hannes Feiler and collaborators. So basically, these optical traits are optically effective plant properties which can be retrieved from canopy reflectance. So these correspond to leaf biochemical content, leading to light absorption in specific spectral regions, such as pigments, water. And also leaf and canopy architectures are responsible for light scattering. So this includes leaf structure as well as leaf orientation, angle distribution in the canopy, leaf area index, and so on. So these are intrinsic properties of vegetation, and in addition to these intrinsic properties, canopy reflectance is also influenced by multiple factors which are extrinsic. So just to give a view for people who are not familiar with optical traits, so these optical traits are basically the continuous alternative of optical types which correspond to optically distinguishable functional types, so which were introduced by Austin and Gaiman. And so why are these optical traits so interesting? So currently, there's an increasing availability of imaging spectroscopy data, which is actually extremely relevant to have a better estimation of these chemical constituents. So basically, we have a full measurement of a continuous spectrum. And so this allows extending the list of optical traits accessible remotely. So we expand the range of type of pigments we can access compared to multispectral data. We can have access to vegetation protein content and many other constituents which were so far inaccessible using satellite data. And so there are a bunch of new applications taking advantage of these optical traits. So here I mentioned a few satellite missions. So you certainly are familiar with the PRISMA, EnMAP, thesis, and the future missions like CHIME from European Space Agency, SBG from NASA, and biodiversity KISP, which is a French Space Agency project. So we can use optical properties in order to also get information about ecological ecological information. So basically, there's a link between optical properties, structure, function, and composition through some chemical fingerprint, which can be translated in optical properties. So this figure shows the chemical fingerprint of a set of of a species collected in a tropical ecosystems. So here is on the left side, it's standardized chemistry for different constituents, and on the right side, it's a visualization of the leaf reflectance for this species. And so, well, here it's not really explicit, but basically you can relate these chemical properties to the leaf reflectance. And so that's an important point. Of course, it's also a simplification of reality. There are intraspecific chemical and optical variability, which are often overlooked. And so we always need to think about remote sensing analysis through analyzing the potential and the limitations associated with the method due to simplifying hypotheses. So how can we access these optical traits from reflectance data? So basically what we usually want to do is to select methods maximizing the sensitivity of optical traits of interest while minimizing the sensitivity to other factors. And so we can use spectral indices, for example, which are computationally straightforward, but also have a few drawbacks. Or we can also use multivariate approaches or physical modeling. And so I will try to give some brief overview of it later. So basically here, these three families of methods, I will not tell you that some are better than the others. It's as all the time, the choice of the method can be driven by multiple constraints. And at some point, you may prefer one or the other method based on the remote sensing data and ground data, which is available, your computational capacity, or some operational constraints. So here I will give you a brief introduction on physical modeling of vegetation. So I think most of you may be familiar with it, so I will try to be as brief as possible, even if I tend to be quite chatty. And then I will illustrate how these models can be used to understand and analyze optical remote sensing. And then I will finish with two applications taking advantage of optical imagery, which correspond to optical time series analysis to identify bark beetle attacks in spruce forest, and the estimation of biodiversity using various proxies computed from optical data. Okay, so first, this physical perspective. So what do optical measurements tell us about vegetation? So if you start at the leaf scale, as I explained earlier, so leaf optical properties, which correspond to reflectance and transmittance, are directly related to foliar anatomy and foliar chemistry. Then, if you take a step back, so these leaves are integrated in the canopy, and so we need to account for canopy structural and chemical properties, so which correspond to leaf area index and leaf orientation, some clumping effect, and so on. If we want to understand how to scale up from leaf scale to the canopy scale. And then, of course, when you acquire optical data, so we work with pixels, and these pixels are also influenced by soil topography, atmospheric property, and so on. And of course, at the end of the chain, we have a sensor which is specified, so have specific acquisition characteristics, such as spectral properties, spatial resolution, revisit period, and so on. And so basically, if we want to get the most from remote sensing, we need to be aware of all the links between these different scales. So optical signal results from interaction between light and matter at all these scales. And we'll first start with leaf optical properties. So as I mentioned earlier, these leaf optical properties, they depend on chemical and structural properties. Here, it's an illustration of the evolution of leaf reflectance with senescence, so from mature to senescence. As you can see, there's an evolution of the leaf reflectance, which is explained by the differences, the evolution of pigment content with degradation of chlorophylls and carotenoids, increase of brown pigments, and so on. So to get a more quantitative perspective on it, multiple methods have been developed to take advantage of leaf optical properties and link them with leaf chemistry. So here I will focus on models. So these models, which are based on physical description of the light-matter interaction in order to link optical properties with chemical and structural properties. So the most widespread model is the prospect model, which assumes a relatively simple description of the leaf as a stack of homogeneous layers. So the leaf seen here in the middle figure, it's just a stack of homogeneous leaves, which allows to directly simulate leaf directional hemispherical reflectance and transmittance from a limited set of inputs corresponding to chemical constituents and foliar structure. So there were successive versions of prospect accounting for an increasing number of chemical constituents. So here I'm giving an overview of the different versions of prospect and the different constituents which were added with these versions. You can find some of these versions in the prospect R package, which is the link is showed here. And so there are also other leaf models which exist, like FluSpect, which accounts for fluorescence, Liberty, DLM, FastSpect, which all have their specificities. And then at some point, you can couple prospect or leaf model with canopy model. So we can perform physical modeling at canopy scale, either using a simple 1D canopy reflectance model, such as the sail model, or using more complex 3D models and taking into account the 3D structure of vegetation. So here it's just an example taken from a work from one of my PhD students, David Bengo, who used some LiDAR data in order to simulate imaging spectroscopy data over tropical forest. So as for the leaf models, a lot of canopy models exist as well. And so what's interesting here is that we have increasing possibilities to integrate a variety of leaf traits, canopy structural properties into realistic simulation for complex vegetation layer. So here I mentioned, so how can we use models in order to simulate data? So we can use it to understand the link between vegetation biophysical properties and optical properties. But we can also use it in inverse mode. So basically predict vegetation biophysical properties from optical measurements. And so this model inversion can be done at leaf scale or canopy scale. And you have various approaches, various modalities to perform this inversion. Basically, at the leaf scale, it consists in estimating chemical constituents and foliar structure based on directional hemispherical reflectance and transmittance. So here is just an overview of different results obtained with the prospect model. So prospect D, which allows for relatively accurate estimation of different pigment, foliar pigment contents, chlorophyll, carotenoids, and anthocyanins. We can also estimate equivalent water thickness and leaf mass per area. So the figure below shows prospect inversion on various datasets acquired in temperate, tropical, and boreal systems. So it works quite well for diversity of vegetation types. Of course, Prospect is relatively simple and it doesn't account for leaf asymmetry and surface effects. So you can always find vegetation types where it may not work, but overall, it's relatively, it works quite well. And then the latest version, Prospect Pro, allows estimation of leaf nitrogen content based on the protein content absorption. So as I mentioned, you can find these models, inversion routines, and a bunch of other functions in the Prospect package. So it's a R package here. And at the canopy scale, so we'll try to be brief on that. I guess the Senseco community is quite aware of this because of Katia Berger and Johan Verhelst, who are active members of the community and specialists on this. So basically, there's a variety of methods which allow retrieval of vegetation properties from canopy reflectance. So this includes data-driven approaches, inversion of radiative transfer models, and certainly the most widespread currently, which are hybrid approaches, which take advantage of physical modeling and machine learning for the estimation of vegetation properties. So, as I said, you can use different types of physical models. So here I'm just giving a brief illustration with the ProSail model. So you can apply it, for example, with Sentinel-2 data in order to estimate leaf area index, chlorophyll content, water content, leaf mass per area, or with imaging spectroscopy, for example, for extended number of properties. Of course, you need to still think about uncertainty in the retrieval of these properties. And this uncertainty strongly varies with the sensor you're using, the vegetation type, the biophysical properties. And so if you want to give it a try, you can find some all the models and inversion routines in this R package dedicated to ProSail, for example. And this is what you can get from applying some hybrid inversion on Sentinel-2 data. So basically, you can produce maps of leaf area index, equivalent water thickness, leaf chlorophyll content, and so on. So at the canopy scale, as I mentioned, you can go a bit further and integrate some like canopy heterogeneity in order, for example, to produce more comprehensive data sets that you will use with machine learning algorithms. So it allows taking into account that kind of heterogeneity that you cannot simulate with more simple models, such as the SAIL model. And so this is also particularly useful to prepare future satellite mission. And so the DART model, more specifically, allows simulation of LiDAR data, fluorescence, thermal infrared as well. So it's quite a comprehensive model. So, okay, to end up with the physical modeling. So physical modeling inversion has strong potential for the estimation of optical traits, and it extends possibilities of multi and hyperspectral analysis compared to spectral indices, for example. It's already integrated in commercial decision tools for precision agriculture, for example, in order to get information about fertilizer intake, yield prediction, drought nutrient stress, and so on. However, at some point, it currently lacks maturity for routine global production. So if I'm in the perspective of ecological applications, so in the ecology community, essential biodiversity variables are quite important variables that we focus on, and some of them can be estimated with remote sensing. And these vegetation traits would be like a very good added value for this framework. So to get there, we would need some extensive open documented validation data from ground data, remote sensing data, in order to consolidate like the model inversions we can get. I mentioned spaceborne imaging spectroscopy. So right now it's in its infancy, but we'll get more and more data to contribute to it. Oops, sorry. And basically, we also need to work on improving the models, improving the account for uncertainty in order to get towards some operational products taking advantage of physical modeling. And so there are also some constraints which may limit the use of physical modeling to estimate vegetation properties, which include the lack of validation data, exploration of large volumes of data, and some conditions which may fall out of the domain of validity of the models. So, for example, for one application, it's the application of process inversions on conifers or very heterogeneous forest ecosystems. And so I will show you these two types of applications and how we can still use physical modeling at some point to contribute to these applications. So the first application I wanted to talk about is the detection of bark beetle outbreaks on spruce forest, which is a work we are doing in collaboration with Raphaël Dutrieux, Kenji Ossé, and Florian de Boissieu in the TETIS research unit. So to give a bit of context, we have outbreaks of bark beetles, which are amplified by climate change and forest management. And there's a strong need for operational outbreak monitoring system currently, because this bark beetle crisis currently is impacting Europe. And so it has strong, including strong economical impacts, for example. So basically, in order to produce these monitoring systems, we identify a certain number of requirements from optical remote sensing data. So as outbreaks do not occur simultaneously, we need at some point some continuous time series analysis. We need to be able to work at regional, national scale, and we need some ways to get detection as early as possible. And so previous work showed that water stress and deficit in canopy foliage were the main symptoms when bark beetles attack. So basically, we decided to focus on Sentinel-2 time series analysis because it has a very frequent revisit. The spatial resolution is compatible with the type of phenomenon we focused on, and it includes a multispectral information with the near and shortwave infrared data which are sensitive to water content. And so how can we detect vegetation water content from Sentinel-2 on spruce forest? So as I mentioned earlier, there's a limitation because Prospect is designed to simulate broadleaf optical properties, while spruce have needles. And so the direct inversion of profile may lead to biased or inaccurate estimation. And so as we are focusing on spectral, so time series analysis, spectral indices at some point were more appropriate as we don't have operational production of biophysical properties for image time series. And so here what we did, we took advantage of physical modeling in order to identify spectral index maximizing the sensitivity to equivalent water thickness, assuming that at some point, even if process is not necessarily particularly appropriate for spruce forest, these results could transfer to conifers. So we tested different spectral indices. Two of them you may be familiar with, NDWA1 and 2, based on near-infrared and shortwave infrared data, so spectral bands in Sentinel-2, and the SEER sphere, which is a continuum removal over the sphere domain, which is illustrated here. I can give you additional details if you want later. And so basically, our analysis resulted in showing that the SEER sphere was more correlated with equivalent water thickness than NDWI1 and 2. So we decided to use it as an indicator for the next steps of our method. So here's just a quick illustration of RGB data over so spruce forest, which was attacked by bark beetles, and the CR3 and near 3.1, 3.2 image. So how did we manage to, so what are the ingredients for our method? So basically here, we analyze Sentinel-2 time series to identify bark beetles, and we perform some anomaly detection in a seasonality of CR3. So basically, it's based on three main steps. So first, we compute the cloud mask and CR3 index from bottom of atmosphere reflectance. Then we adjust a harmonic model for each pixel, and this harmonic model is based on a training period of 2 years. And then for the following years, we detect anomalies and a confidence index. So basically, we only need Sentinel-2 time series and a forest mask, and it's a pixel-based process. And then we can obtain at the end some raster and vectors corresponding to the detection. So here at the bottom is just an illustration of seasonality for a healthy pixel, seasonality of CR3 for healthy pixel on the left, and a pixel attacked by bark beetle on the right. So you can see there's clearly a strong effect of bark beetle attack on the CR3, and so that's why that's what we took advantage of. So what can you do with this method? You can produce some animated images. Here it's just an illustration of the evolution of forest dieback with time. And basically, these are the two main products. We get a detection map, which shows when pixels were attacked, and an anomaly class, so which gives some low and high anomalies, which is basically some kind of confidence index. And so you can find this method and all the documentation and tutorials in the repository following this link. So this was developed by Raphaël Dutrieux, Tethys Research Unit. So I will not go through this. And so basically, then we used it at regional scale in France in order to monitor bark beetle attacks over different periods of from 2017 to 2022. And here on the left, you can see the yearly estimation of bark beetle outbreaks. And on the right, it's accumulated surfaces of spruce suffering from dieback from 2018 to 2022. So we try to get this as operational as possible. And hopefully at some point we can compare it with other methods developed by colleagues from Europe. So this will be the next step. And so I will try to quickly end with spectral diversity metrics, which can be used as proxy for biological diversity. So this is another context, it's more an ecological context. And so here I will try to explain you how we can go from remotely sensed information to ecological information. So how can we link reflectance and optical trace to ecological information? So here, everything I will show you will be based on the attempt to identify a relationship between spatial heterogeneity of spectral information and some environmental or ecological heterogeneity related to biodiversity. So what can we use to approximate optical traits? So here I will mention a few approaches. So you can use either transformed reflectance as shown, so mentioned in the two references here, or spectral indices, for example, or biophysical properties, which can be used as proxy for optical traits. And then you need to express these spectral variations using either univariate, multivariate space, discrete or continuous space. And then at some point, you need to identify which ground observation you should compare with spectral information. So it depends whether you're interested in species or taxonomic diversity or in functional diversity, for example. So it's always a matter of selecting the right data, the right methods, right hypothesis to match with like your object of interest. So here it's I'm detailing here the type of information you can use to approximate optical traits. So you can either use transform reflectance, so which has no prior assumption on thematic relevance of spectral information. Usually it's based on data-driven approaches, which is possibly limiting when you're studying sites independently. And it can be based on manual automated feature selection. For example, you can perform principal component analysis to transform your data and to help reduce sensor noise and artifacts, for example. So all of this is explained in the publication here, the two publications here as reference. And so basically to illustrate it, you can, if you get, for example, a Sentinel-2 image from tropical forest in Amazonian Peru, here, you can perform the different processing steps in order to get 3 components here that we select for display, which allow identifying different types of vegetation types. So this can be done in a relatively simple way, and then you can use it to analyze further your data. You can also use some spectral indices, and so that's the approach which was developed by Fabian Schneider, for example. And then basically you select spectral indices based on their correlation with vegetation indices. And then you try to identify so some diversity metrics based on this combination of traits. Here it's an example. So using the same Sentinel-2 image, we computed different spectral indices and we obtained this color composite for the same forest. So as previously, you can see some patterns corresponding to different vegetation types. So still nice colored images. And then you can also use biophysical properties, basically by using Procell inversion here in order to estimate vegetation biophysical properties. So it's computationally more intensive, but it's also easier to interpret by ecologists or agronomists when you discuss about leaf chemistry or leaf area index rather than some obscure reflectance or spectral indices. And so if you apply physical model inversion on the same site, here you can see also a color composite you can obtain based on leaf area index, chlorophyll content, equivalent water thickness. And so it's just these 3 approaches. It's just to illustrate that you can basically at some point select different methods in order to produce higher level metrics aggregating spectral information. And then at some point, if you want to move from there to ecological information, so one possibility is to define a spatial extent where you will work and basically integrate the heterogeneity over the spatial extent. So for forest ecosystems, for example, we usually work with 0.25 or 1 hectare that like plots, which usually match with forest inventories. And so, well, I will go very fast through it. So as you can, there are multiple modalities to then take advantage of these proxies for optical traits in order to produce some diversity metrics. So here I will just show you an illustration using a method that I developed, which is based on the analysis of discrete feature space to estimate taxonomic inventory metrics. So basically, here we will use optical imagery, hyperspectral, multispectral imagery in order to estimate alpha diversity and beta diversity metrics. So if you're not familiar, alpha diversity metrics correspond to local diversity, which can be computed at plot scale, and beta diversity metrics correspond to dissimilarities between 2 spatial units. And so how does this work? So here we developed a method which is based on some kind of naive approach as we could see, we could identify in previous work that imaging spectroscopy is relevant to identify tree species. Then we thought, okay, maybe it's possible to just apply some clustering and assume that at some point the clustering will provide some relevant grouping corresponding to species. And so here, what we do is comparing some species inventory with some cluster inventory obtained from image processing. And so we also do it for alpha and for beta diversity. And all of this can be done with a spectral diversity analysis performed with the biodiv mapper package. And so you can find tutorials and all the information you need. So following the link and reading this publication. So basically what it does, it allows linking imaging spectroscopy and multispectral data or multi-source information to some diversity maps. So you can produce these diversity maps. And as I said, you can also use physical traits, data-driven trait estimation in order to get these diversity maps. And so basically here, the systematic use of biophysical properties would have a great added value instead of using transform reflectance as it was done in the first versions, actually, as it may improve the robustness and consistency of estimated diversity metrics over space and time. So that's one very important point for advocating for better operational estimation of vegetation traits. And so maybe I'm a bit too long, so I will go rapidly through the illustration of what you can do. So here it's just a validation of the method. So we performed some inventories over the tropical forest, and we compared the estimated Shannon index with biodiv mapper and the measured Shannon index corresponding to forest inventories. And here, so here, alpha and beta diversity maps are the products you get from biodiv mapper. And so we could see that we had a strong correlation between alpha diversity measured and estimated here. And so we also applied it on temperate forest. So here it's another hyperspectral acquisition from a site in southwest of France. We also had some pretty good correlation between the measured and estimated Shannon index. And here it's the map we obtain, and we can see that the patterns corresponding to pure and mixed forest match with the patterns obtained from the Shannon index map obtained with biodiv mapper. And so this was obtained with imaging spectroscopy, but the good thing is that you can also at some point apply it on Sentinel-2 data, as I showed earlier. So it's also possible. And so here it's just a very brief illustration of a comparison between what we get with biodiv mapper and what was obtained by Chavez and collaborators when they provided a method to map floristic patterns in Peruvian rainforest using some supervised approach with forest inventory data, Landsat data, climate data, soil elevation data. So they use a bunch of information to produce these maps. And here on the left side, it's the map which was produced by during this study. And so I just decided to select a very small subset on one Sentinel-2 tile right here. So I extracted this region. And so here on the right side, so on the left side, you have the Sentinel-2 acquisition. On the right side, you have the reference data obtained by Chavez et al. Based on the supervised approach. And here are the outputs you get from biodiv mapper. So basically here we can see we are able to obtain the same kind of patterns. So actually it's just a comparison of two products. Chavez et al. Validated their map, but it's we still need to go further in the validation, but it's at least it's a good thing to see that some supervised approaches and unsupervised approaches result in identification of similar patterns for that kind of tropical ecosystems. Okay, and now I will finish with my presentation. So in conclusions and perspective, optical traits contribute to bridge the gap between remotely sensed signal and applications on the ground in ecology and agriculture, forest management, and many other fields. So operational production and use of optical traits still require efforts, but we already have a bunch of solutions like softwares, data, which are publicly available, which allow people to use it for their own need, even if it's not a global coverage that we get. So methods and data currently available, the open perspective, as I mentioned. And so the methods based on spectral diversity and optical traits can also contribute to ecological application. But you need to keep in mind that, as I said earlier, we need to work on both potential and limitations of the methods we are developing using remote sensing, and we need to investigate these in a balanced way. Accounting for spatial and temporal dimensions of analysis is key, of course. And the method that I showed you, particularly for biodiversity mapping, certainly need adjustments depending on the type of ecosystem under investigation. You will not work with a forest ecosystem the same way as you work with grasslands, for example. And here are just the reminder of the different packages which have been used to produce the results shown during the presentation. So everything is publicly available and you can access it, use it, change it, do what you want with it. And this is another set of packages which were developed by our research unit, TETIS, in Montpellier. And please feel free to use it and contact us if you have any question when using it. It will be a pleasure to collaborate with you or help you if there's something wrong going with them. Of course, we are not the only ones to develop methods, packages, and so on. And here are a few references for some work done by other colleagues in the field of ecology, for example, and some toolboxes which are very useful. Well, I know you're familiar with Nmapbox, as you had certainly several presentations about it, same for Artmo. So I'm inviting you to explore all of them and have fun with modeling and remote sensing. And thank you for your attention, and I hope I was not too long